minimum which may never be reached because it's not accessible. Now this is a sort of energy landscape which represents various kinds of crystal forms, polymorphs. But we can define a corresponding equivalent for structures themselves. So the landscape will then include polymorphs, co-crystals, salts, everything that is en route to the final crystal. Crystallographers in the audience will appreciate what I'm saying when I say crystal forms with multiple Z prime. So crystal forms with multiple Z prime may be frozen snapshots of an intermediate crystallization process before the thing gets to its final state. I mean, it has to be pretty late in the crystallization to see it as a crystal, because don't forget, a crystal has three-dimensional periodic order and all that. So it's reaching towards the late stages. So this landscape covers essentially the late stages of the crystallization event. And this is the landscape is a thing which we can exploit profitably to make new solid forms. So it suddenly becomes something of extreme practical importance. So being able to access different portions of the landscape and looking at various things here and there is quite important. Polymorphs, etc. Okay, recent example. I just went to one journal, Crystal Growth and Design, and in the last 2 years, there are papers on polymorphs of all these things, and there's probably more if you look in other journals and so on. So there's not an API that people are not looking at polymorphic forms with the idea of suppose generating new polymorphs or whatever. This is in the published literature, so what is proprietary must be. So omeprazole here, what do I mean by designing properties? Now this is a very interesting API. There are two forms, the 5-methoxy and the 6-methoxy. And they have basically the same crystal structure. And the polymorphs of omeprazole differ in the proportion of the 5 and 6-methoxy tautomers. So you start with 100% 6-methoxy, which is one form, and then you can increase the amount of 5-methoxy till you get to about 15%, 15% of the 5-methoxy, and there it stops. Now, there are 3 or 4 forms in the middle. Now, what's the difference? If you look at the pointer pancake level. See, this is the this is the pure 6-methoxy form. You notice it's a layered structure, and then these little red dots here represent the methoxy group which is bent out in the 5-methoxy form. So if you look at the 6-methoxy form, the layers can slide like this, and it is very soft. The moment you put these little red dots, 5-methoxy group, it becomes like a Velcro thing. It becomes, now the layers cannot slide past each other. So you see, even the 0 to 15% 5-methoxy, you can start playing with the mechanical properties of the solid. And this is exactly what Manish did. He's here in the audience. And yes, what he did was he used a technique called nano indentation, where it's literally like poking a crystal with a nano needle, and you can then get an estimate of the hardness. And so you see there's a nice coefficient in the correlation between the amount of the 5-methoxy tautomer and the hardness. This is what crystal engineering is all about. So if you can change the amount of the tautomers at will, you can then change the mechanical properties in a regular manner. These kinds of things can be done for many properties. I just give this as an example. Now, I think the reason why lots of people may be wanting to study co-crystals in genetic pharma is that there was some clarity in the recent guidance for industry by the FDA. I'm sure Steve will be referring to more of this, so I'm going to continue onwards. But shall I say that I found this sentence to be more significant. Pharmaceutical co-crystals have opened up opportunities for engineering solid-state forms conventional solid state forms of an API such as salts and polymers. I think that one sentence has put the whole crystal engineering enterprise really in the foreground. Basically, FDA is saying, okay guys, it's good to work on these things. And the word engineering has been used. So it's a clear message to the pharma industry that this crystal engineering is something that can be profitably exploited. We needed this regulatory clarity because the clarity was not there in the earlier definition of the term that came in 2012 also. Now it's there. It's very clearly written. These are the synthons, and I want to talk about co-crystals, and you can use synthon theory to what I call supramolecular homologation. In other words, increase the number of components. The problem with co-crystals is that things like to crystallize as pure compounds. Crystallization is a purification technique, not a technique where you bring things together. Things come out when you start crystallizing. They don't come together. So when you want to come together, you have to do something specific. That's what you use this synthon. And how do you homologate? Dr. Venkateshwar, I'll take you back to your synthetic days. You know, molecular homologation, there are so many tricks. So supramolecular homologation, you can do it something like this. This Renu dating, he's also here in the audience right there. And all these people got their PhDs with me at various stages and are flourishing elsewhere now. You take this amide here, which is a very standard synthon, and then if you put an acid, a diacid typically, the acid interrupts the amide hydrogen bonding pattern almost always. In other words, the acid amide synthon is much better than the amide synthon. So when you do this, it's a very easy way of adding the number of components by one. That's why I call it supramolecular homologation. So from a one component crystal, I can get a two component crystal. By using the same trick with a two component crystal, I can make a three component crystal. And this is what synthetic chemists like to do all the time. You need a general method which will be very robust and which you can keep applying again and again. So this is a way of making co-crystals with pKa rule again. This rule of 3. So if the pKa between acid and base is more than 3, then we get a salt. If it is less, it is less. Arijit is here, he did this work. This guy was running around with me with the bag. And then this is a very recent paper. I must tell you to please go to the booth. IUCRJ is our new journal, open access journal, the flagship journal of our union. And this was a wonderful collaboration we did with people in the Geol company in Japan. We used 1100 megahertz solid state NMR to look at this natural abundance N15 was the technique used to find out the proton position, because as we all know, X-ray crystallography is a very poor way of finding a hydrogen atom position, and yet for the salt co-crystal problem, it is the precise location of the hydrogen that is most important. I can stop now. This is about all that I wanted to say. These are the things that I'm sure the other people will speak about. Many of the drugs which are all blocked are in this class 2. Class 3, class 4, everything has to be pushed up to class 1. So there is a permeability issue, there is a solubility issue. I'll just state the problem briefly. Maybe Professor Nandya will talk about it. Somebody should say something. The chemical factors needed for both of these are contradictory. The more ionic it is, the more soluble it will be, but the less will be the permeability. And if you try to make it more, what shall I say, lipophilic, then the permeability will increase, the solubility will come down. So there must be some golden region in the middle where you can optimize both these things. And I believe permeability optimization is one of the key issues in solid forms. And we cannot, for so many years we have only been worried about the solubility, not caring about permeability at all. So this is another issue. Yeah, this is Nangia's model. Maybe he'll speak about it something. This is permeability, hydrochlorothiazide. We tried, we were linking permeability to synthon structure. Yes, that's what we did here. Where is Palaj? Palaj also maybe better drug in these aspects. So there is a formation of an ionic co-crystal between these two. Prasad, who did this nice work, which was covered in the Hindu newspaper also, rather extensively. When you make this salt co-crystal, then the permeability goes up. And you see, essentially what you're doing here is, this is the salt co-crystal hydrated. So there is some synthon-like intermediate in solution, so that the sulfur thiazole is able to drag the norfloxacin through the membrane. And this won't happen in a physical mixture of the two. So when in the physical mixture, when you try to, actually we see norfloxacin physically on the membrane, not able to go through. And I will not steal Professor Nangia's thunder tomorrow. He's supposed to give a keynote, so I'll skip this example. And but that's it. So the modifications of physicochemical properties of drugs through new solid forms are an area of importance. CE provides useful information on the mechanical properties of polymorphic drugs, which in turn allows for developing an understanding of stability. The principles can be used to achieve these properties. So you are linking structure and property through syntons and other things, without influencing the therapeutic efficiency, of course. So the idea of the co-former, it has to be a gas compound, otherwise you're wasting your time. And then SSNMR, I believe, is a technique of the future, especially when it concerns the salt co-crystal conundrum. Because the more you look at these X-ray structures, neutron is not a practicable thing. So you need big crystals, you have to go here and there to various places. You can't have neutron in a lab. So it is not a technique that we can do. Solid state NMR is really. Right now only the Geol company has a direct temperature, but I think they're going to start selling it to us at a very high price before long. So we will get a hold of it. So this is about really what I wanted to say, and I leave it for an open discussion, or maybe the questions will come out through the other people. It depends on what you all want. Which way shall we do it? Steve, will you say something about the regulatory aspects and the solid forms first? Maybe we have 2 lectures, because the other things now concern more the industry part itself. So I think it's better for the audience even see if they listen to the more academic types of talks first. I think that's then maybe you can even ask us questions together and stuff like that. Yeah. So okay, Steve, from the floor is yours.
Okay, thanks, Gautam. It's a great pleasure to be here, and thanks very much for inviting me. I'm going to, I've actually worked in all 3 areas in the FDA, both as on the Pharmaceutical Science Advisory employee and also in academia. And then my wife and I started a company called Solid State Chemical Information. And we started with 2 people and it grew to 100. We sold our company in 2006 because I'm really a professor at heart. And she was worn out with running a company. So, but we really enjoyed it and it was fun for us. So I'm going to talk about the regulatory aspects and how they interface with academia and solid state area. And so I used a simple Venn diagram. There's an area where regulatory science, and I'll define that in a little bit, and solid state chemistry overlap. We could debate. It could be bigger than this, but certainly there's a major area of overlap. And it actually started back when Gautam, just a little bit before Gautam came to Illinois. What year was that? 72. Okay, so 69, Gautam came to Illinois and I was leaving and he took my desk. So we go way back. And so there was a paper in 69 and I'll talk about that started a lot of the regulatory. I'll talk about a number of books. There's the so-called generic drug scandal in the US. And that actually has a solid state aspect. In fact, a lot of it was solid state. There's the famous ritonavir, and then Gautam talked a little bit about enabled formulations, amorphous formulation. Whoops. I can turn that back on. Okay, so this is the 69 paper by Brenner. Brenner was a Merck, and they observed that they had a tablet on the shelf, and as it stood on the shelf, if it was in this form, this A form, which had water and ethanol in it, it oxidized to 72% in 2.5 days. If you had this C form, and the A form was obviously a solvate hydrate system, the C form, no oxidation whatsoever. So this laid the groundwork for regulatory issues in solid state. You could have one form that would be changed, and you'd have another solid form that remained stable. So this started the whole field. And DY, and I'm sure he told Gautam this too, but DY said, well, Steve, you're going to a school of pharmacy, you should work on some of this. This is a good area for you to work on. And so that's sort of how we got started in this area. And then it actually took until around the mid-1980s, 84 to be exact, when the Generic Drug Act passed in the US. And this is called it's really the Hatch-Waxman Act, but it allowed copies of marketed drugs to come on the market. And maybe it's better if I go to the next slide and then come back. And here's a report in 1987 of a 30-year-old epileptic woman who had seizures, serious seizures, that were well controlled with the brand drug. When she started on the generic, the blood levels of the drug dropped dramatically. And what happened after a series of investigations is the following. The generic product contained the dihydrate form. The marketed product contained the anhydrate form. In fact, even though the tablets had a lot of moisture in them, around 8%, it didn't convert the API to the dihydrate. The anhydrate, as we know generally, had higher solubility and reached higher blood levels. The dihydrate, lower solubility, lower blood levels, less therapeutic action. So this was like a clinical trial of a soluble and an insoluble solid form. It wasn't done intentionally, but that was the effect of it. And it had a huge effect on the FDA. So the FDA started to become quite interested in this area. And so this is the company that I mentioned that we started. The date here is 91. And this is a short course that we decided to teach in New Jersey with myself and an industry partner named Ralph Pfeiffer from Eli Lilly. Ralph was a powdered crystallographer from many years prior. And actually, it would be great to dedicate this talk to Ralph. Ralph passed away about a year ago, and he would have loved to have been here. So this course was a 2-day course where we started discussing solid forms, control of solid forms, and the potential effect on pharmaceutical outcome. And at this course, sitting in the front row, was a new FDA scientist named Mike Gainey. Mike was an MIT PhD who had worked at McKinsey, a famous consulting firm in the US, and he started asking me about developing flowcharts and decision trees in solid-state chemistry, and how these could be used to help the FDA. And I'll get to that story in a minute. Back to the introduction now. So then in '98, the famous ritonavir system came on the scene. And ritonavir is marketed as Formed 1 at that time. It was dissolved in a soft gel capsule, and it was curing AIDS. It's the Magic Johnson drug. I'm sure, I believe anyway, that Magic still takes the drug. And it was a protease inhibitor, and it was the magic drug for hundreds of thousands of patients. And it started failing dissolution. It was in a soft gel capsule, so it was dissolved. It started failing dissolution, and a new form was observed, as you can see from the powder diffraction here, and it was Form 2. Pardon me, Form 2 is these spherulites, beautiful crystalline spherulites. And Form 2, in the dosage form, had about half the solubility as Form 1. So you could isolate from the soft gel capsules crystals of Form 2, and that meant that the drug didn't dissolve because it crystallized in the tablet. That meant the blood levels were not high enough in the patients to combat the AIDS virus. And so AbbVie, I still remember when they called me, it was the spring of '98. I was driving to Purdue, and I was so amazed by this story that I pulled off the road and grabbed my laptop and started making notes. And they said, well, would you be willing to consult? And I said, of course. And I said, when would you like me to come? And they said, this weekend. We're having a meeting this weekend to try to solve this problem. And set off one of the longest projects that I've worked on. The estimated cost to overcome the problem, including lost sales, was $900 million. And they had to write all of the AIDS patients that were on this drug and tell them they were going to have to withdraw from the market and switch them to a solution formulation. The crystals of the drug are brick dust, so they don't dissolve. They're not soluble, and I'll show you. And the liquid formulation tasted so bad that many of the patients said, I'd rather die of AIDS than take this formulation for the rest of my life. So you had a year there where the drug was being reformulated, that it was off the market. And here's a picture of what you would call the enabled formulation, the problems with the crystals. The crystalline formulation, this is blood level versus hours. The crystalline formulation is this little bump right here, essentially no blood level. The amorphous or dissolved formulation, different ones, obviously reach much better blood levels and are much more bioavailable. So this is the story and the goal of solid-state chemistry, as I'll get into in the pharmaceutical area, is to increase solubility, increase bioavailability, same with co-crystals, same with salts. All of these strategies involve using the solid state to try to achieve better solubility and better therapy. And so, because of that, and these next two slides, we're just going to talk about this in more detail, because of that, solid-state chemistry has become extremely important to the FDA. And let's go back now and talk about the other side of the Venn diagram, the regulatory side. So regulatory science, some people say it's a contradiction, but in fact, it involves developing new tools, standards, and approaches. So the co-crystal strategy would fall into a new approach. The tools strategy would fall into, for example, solid-state NMR and paradistribution function. Those are new tools. And academia typically focus on the tools and the approaches. So that's what we do in academia. Fundamental understanding of tools, I'll say NMR and so on. Fundamental understanding of approaches such as co-crystals. But all of these studies directly bear on what you would call safety and efficacy. How the drug works, how safe it is, and how efficient it is. And so it directly bears on the quality of life people achieve that are taking a drug, that have a disease that you're trying to cure or attack and minimize. And the way you would say this is the safety of a drug and how efficient it is, whether it works or not, is directly related to dose, how much of a blood level you can get. And the dose is related to solubility, as Gautam was showing, and solubility is related to the solid form. So it's a two-step process. Solid form controls solubility, solubility controls dose. Now, also, as Gautam was talking about, we have permeability coming in there, which is another whole factor. And the BCS classification system that Gautam showed deals with this area. So do the BE waivers. And here my number was 80%, and Gautam showed a number of 60%. But somewhere between 60 and 80% of the drugs on the market.
As high as 80.
Yeah, I think it might be higher than 60. Certainly, for example, the new hepatitis C antiviral drugs. These drugs are amazing drugs.
If I can interrupt, Steve, because
yes,
I think the reason this number is so high is because some of these new molecules are they try to discover them in silico.
Exactly.
And I think these computers, they are not gods. You know, sometimes I really feel that old method of discovering drugs had a lot to do with it. I mean, I've read an article on why did they put the nitro group in that why did they put the nitro group in ranitidine? And it's a wonderful example of qualitative thinking. The guy simply said, oh, I think if I put the nitro group, the solubility will go up. Now he was correct. And that's how drugs were discovered. Sorry to be nostalgic, but I think it is the silico business that has led to this 80%. So, you get to this problem. Sorry to interrupt.
But this poor solubility makes a lot of jobs for solid-state chemists. A lot of jobs for you guys are created by poor solubility drugs, so it's not all bad. And the new hepatitis C products, those are all soluble as brick dust. Those are all very difficult to deal with. And they're curing people. They're walk-away cures, which most of us, we would have never thought you could cure a virus with a drug. We would have thought you had to have a vaccine that prevented the infection in the first place. So it's a pretty big advance. And a number of the cancer drugs also fall into this very poor solubility. Another way to put this is the poor solubility means that the solid-state chemists or the crystal engineer have to come in and engineer something or change something in order to get good solubility. Otherwise, just like the ritonavir case, patients will take it, it'll go right through this system and come out with feces. So it's a pretty practical problem of improving solid-state chemistry. And I think we can just skip this slide. We've basically been covering this. There's a very interesting emerging concept at the FDA on equivalence. Now we're going to put a generic drug ad on. We're working at a generic company. We want to develop a new product. What do we have to do to match the innovator product that's out there? Well, certainly, according to the concept, they have to have the same components. Number two concept now, same concentration. That's the same amount. That's not typically an issue. And then the most amazing third issue same arrangement of matter, same microstructure, which would mean same polymorphic form. This would, there have been historical instances where two different polymorphs that had roughly the same solubility were approved as a generic copy. What this is saying now is we're going to have the exact same polymorph. Anytime we have new forms, that's a new medicine, and that's the opportunity that Gautam was talking about, the opportunity to make a new medicine which is different and has possibly more bioavailability, can treat more patients on the market. Now, all of this led to these famous decision trees, and I was telling you, just to go back, about the scientist who was sitting in the front row that thought it might be possible to make decision trees. And this is the paper that we wrote, and this is the first decision tree we developed in the polymorphism area, working with 3 scientists from the FDA. So this is a true academic, industrial, academic government collaboration. The first 2 authors are myself and Ralph Pfeiffer from Purdue. The last 3 authors were FDA scientists. And all 3 of those scientists now are vice presidents at their various companies. And the decision trees were taken into the International Committee of Harmonization guidelines for specification. And these guidelines are called the ICH-Q6 document, Q6A. This is the document from the International Committee of Harmonization that really governs worldwide how we deal with solid forms. And notice they list a number of properties here that you would think were important, but impurities we know are important and can be fatal. Polymorphic forms are right up there. So these are some of the main properties that we're concerned with. In the decision trees, they tell you over here, how many people have seen these trees? Just asking. Just a couple, about 5 or 6, yeah. The decision trees have a bunch of directions about how to deal with a polymorphism, and I don't know that I should use time going through all these, but I could just go through quickly and say, here it tells you to do a polymorph screen and figure out whether different polymorphs exist or not. So this is another job-producing system. This is why we have a lot of jobs in this area, because the FDA has told us, told the world, and now the ICH has told the world, to do a polymorph screen, find out how many polymorphs there are. And the FDA term, incidentally, when they use the word polymorph, they're also talking about hydrates, amorphous forms. They're not just talking about unsolidated forms. They have a different definition of polymorphs. And then screen 2 says, do the polymorphs have different properties? And this would be different forms. We really would say, do the different solid forms have different properties? And then if they do, we're told to either set acceptance criteria, which brings in the analytical, what percent of the polymorph must be present to be accepted. Or we have to go to a third question where we go to the drug product, the medicine, and we're told to monitor the form during stability and make sure that there's no conversion to other forms that we don't know the bioavailability. So the whole goal here is to maintain bioavailability. And this is just outlining what strategies have developed in the industry. Yes?
So I have one question about this bioavailability. Do you understand me all? Yeah. So when the last aim is to have the same bioavailability, why, to begin with, we say we have to have the same concentration? When I can make something which has double bioavailability, why can I not take this double bioavailability and have half
So the main concern is not to have too low a bioavailability. Okay, but if you can make a product that has higher bioavailability, is that what you're asking?
No, I'm asking, so we want to have bioavailability.
Right.
But the first question is, when I have a 100 mg drug product, I
A
have
dose.
to have a 100 mg drug product. Why can I not say, I increase the bioavailability, and I go for a 50 mg drug product?
Sure. So this is a big area that we're quite interested in. So you want to make what he's describing as a reduced dose product.
Yeah. With the
A
same
reduced
bioavailability.
dose product has a huge advantage.
Yeah.
There's less drug and it costs less. Especially in the generic area where the API is the most expensive. So if we could halve the amount of API and achieve the same blood level, we could achieve a tremendous cost advantage. The the law required the same dose, but there's a loophole, and it's called the 505(b)(2). And what's called, anybody ever heard of a 505(b)(2)? Yeah, and these guys know a whole lot in this field. So a 505(b)(2) allows you to submit a new drug application. Now, not a ANDA, but an NDA. But you can still use the clinical data of the NDA that had the original drug in it, and you do just what you said. You match the blood level, so you say, I've got a lower dose drug, but I have the same blood level. And those are extremely important products. And there are famous stories about these products. So this is one of the areas that the generic drug industry in India can operate and has been operating. So this is a very exciting area, really. In fact, I don't want to go up too far, but Teva, there was an innovator product, the dihydrate of azithromycin that Teva had on the market. A generic company, Pfizer had the dihydrate on the market. Teva, generic company in Israel, figured out how to make a monoethanolate that was bioequivalent, but didn't infringe the dihydrate patents. They were able to come on the market with that product. There were only 2 products on the market. A rough estimate of the profits that Teva got from that was $2 to $3 billion. So that's another area where generic firms can operate and solid-state chemists can operate, is to make equivalent polymorphs, but unpatented. Equivalent but new, or it could be a co-crystal. Equivalent but uncovered, and cover it yourself, and then you would have a product that would be competitive with the product on the market. So this is talking a little bit about solubility. I think probably I've used up enough time, and I should probably finish up. Let me say one more thing. This is in the quality area. Polymorphism has become extremely important. This term here is critical quality attributes, and right up here we're listing polymorphic form. So polymorphism is very important as a critical quality attribute, and this is my last slide. Polymorphism is also very important in this most difficult area called process validation. So regulatory agencies require you to make a product in a validated way. It works the same way every time. Again, polymorphism and how you control that is absolutely critical. So just to finish up, we covered historical, a little bit about modern regulatory, this equivalence concept, polymorphism, and process validation.
Thank you. Very clear exposition. I'm sure there are comments and questions. Yes.
Steve, a question about flow chart. The first diamond is, have you done polymorph screen? So what is, what is a good polymorph screen? I get that question very often whenever I use this graph. Okay, how much polymorph screen you did? Is it enough? Do you have a standard for it? You have to do 100 experiments or 2,000 experiments in high throughput mode?
So I think so that question is, what is an upper polymorph screen? You could do hundreds of thousands of experiments, crystallization. If you remember back to your organic chemistry lab, you were all crystallizing a material. Some people were getting it right away, some people didn't get it at all. And there were all these different nuances that were used. We usually think about it in three ways. This is the way we did it at SSCI. When a drug was under development, we did what's called an abbreviated screen with about 8 to 10 solvents. Then we did a comprehensive screen when the drug was close to the market. And then we did a heroic or scorched earth screen, okay? When the drug was making billions of dollars and firms wanted to know all the forms, the whole landscape that Gautam showed. You want to know every nook and cranny in that landscape if you have a huge product. So I think I don't know, but it seems like the landscape, the first one you just want a few, and really you're just trying to get on the market and get it in a patient, see if there's any response, and see if you can cure a disease. And then the next stage, you want to know pretty much the important ones, and the landscape is a very good drawing for that.
I would answer your question in a slightly different qualitative and more Indian way. When I am asked to search for all the polymorphs, I would, for an API, I would say that in the hands of one student who I trust, 6 months of work will probably give me all the polymorphs I need to know, whether they exist or they don't exist. Yeah. But as you can see, it's a very subjective sort of decision, but I view it more in terms of time. The time spent in the hands of somebody whom I trust. So, I mean, I can't define that, or FDA can't use something like that. But it works. It works.
That's probably a comprehensive, it's certainly not an abbreviated screen. That's a Neumann, one of Neumann's people, I don't know where you know this name, Neumann. There's a computational scientist named Marcus Neumann. He got every compound in the Cambridge crystallographic database open polymorph competition. He has the equivalent of 200 laptops hooked together and it takes over a month to do a full study. What was very interesting is some companies have been afraid of the ritonavir problem, a lurking polymorph out there that's really stable. Because if that happens to those companies, they're going to lose $900 million, probably even more today. So he showed some data, didn't tell what compound, but they ran 30 compounds. They had two, just kind of supporting what Gautam said. They had two that there was a low energy polymorph that they hadn't found yet. But most of the rest of them they found. So it's pretty interesting, and I'm sure those executives aren't sleeping well, the ones that have those two.
Yeah, I know Marcus quite well, and he's always right, but the problem with him is the extreme cost of the extreme cost. It goes beyond the paying capability of most companies.
Exactly.
But it always works. He won't tell us what it is, so he won't publish it.
They're reparameterized. Okay. So they write a new parameter for each compound, but they won't tell how they do it, and they won't tell what the parameter is.
They don't tell anything.
And in this lecture, they said, well, we're just going to skip over that. So they were pretty good.
Yes, Ashwini.
If I understood it right, after reading the co-crystal FDA guidance, 1100 megahertz, solid state and our classification of co-crystals.
It's not mine, geology. I can't afford it. Anyway, I'm sure there are going to be more questions for Steve during the rest of the symposium. And before you go, I can't but resist. I mean, he made some very quick references to the same desk and somebody called D.Y. And I don't think most of you understood, and most people don't even know who my PhD supervisor was. But his name was D.Y. Curtin, and he was also Steve's supervisor, so I am his junior in the lab. And this is the famous D.Y. Curtin of the Curtin-Hammett principle. So I would like to publicly acknowledge this great man today. We've never shared a podium before, never.
Yeah.
I mean, we've seen each other so often, but
Well, you can tell we think a lot.
but it's the first time the two of, he always calls him D.Y., I always call him Dr. Curtin.
Give me a pose.
Yeah.
Smile,
Well, we should dedicate this symposium.
I think maybe in a sort of way, and he passed away 6 years ago. Exactly, I remember during the Madrid Congress, and I got the news of that when I was in Madrid at the grand old age of 92, and we were some of his last students. He had something like close to 100 students, which was really, really high, and one of the founding fathers of physical organic chemistry. And so we were practically at the very end, But anyway, it's nice to think about Dr. Curtin now. Now look, people, I have to step out briefly for some press conference or something. So the 15 minutes thing will be given by my colleague, Professor Nangia. And we're staying on the topic of government. And no better person than him, because at the same time, he's both academic and government. I don't know how he does it. What I would request you, I wanted to know whether professors become doctors when they go to the national labs, or does a professor always stay a professor? I don't know. We'll have to call we have a horrible habit when directors want to become professors and professors want to become directors. This is the problem with science in our country. But anyhow, we'll speak for about 15 minutes, and if I don't come back from the press conference, please introduce. I've already introduced him, Dr. Venkateshwar. He'll take over and come back.
Thank you. Good afternoon, everybody. As was mentioned by Professor Desiraju, I am more of a fill-in. My boss, Dr. Girish Sahni, who heads the CSIR labs in India, fell ill last night, and he was supposed to be speaking at this time. And in place of him, I will try to fill in the gap to cover the topic of government, academia, industry, interactions in the area of drug discovery, the possibility of what would be a good research collaboration funding model. I should add here that Dr. Brahaspati, who is here, who was Secretary of SERB, all of you know about SERB from yesterday evening session. During his time, SERB did come up with several innovative platforms to bring together industry and academia to collaborate. So what I will do, I don't have any slides since I was informed this morning, and therefore rather than make it into a scientific lecture, I thought I will make it part informative and part autobiographical, and share with you what my experiences have been. As was just mentioned, in academia for about 27, 28 years, I still consider myself as being part of the academic system at University of Hyderabad, which is no more than 2 or 3 kilometers from here. For the last 18 or so months, I have been at the National Chemical Laboratory in Pune. And the third is industry, taking the lead of Dr. A.V. Ramarao, who, to my knowledge, is the first scientist in the country who started his own industry after superannuation. I have a small startup company which is based at University of Hyderabad. There is a technology business incubation cell, and this company I started about 5 years ago in 2012. So in some ways, in parts at least, I have like a Venn diagram. I in some ways represent academia with a large circle, government or the CSIR lab system with a slightly smaller circle, and the academic with a circle of about the same size. I think in the last 30 years, I started my career at University of Hyderabad in 1989. So broadly speaking, one can say it's about 3 decades. When I started in the early 90s, there was funding available. There was DST at that time, and Dr. Brahaspati was in DST in those days. We used to get limited funding. There was no big money, but there was enough money. I always use the analogy of a middle-class family. There's always money for the right needs, but rarely for luxury. We got money to do the research, but it was hard to get money to start a new area of research. So in that sense, it was different from the present model. So typically, you joined a department, and after joining, you made an assessment as to what area of work, if you started a program in, you would have at least the basic infrastructure in place, and add on to it additional research areas which you are also interested in. And I think this model continued in India till about 2000, for about 10 years of my own career, professional career. We went through a dip in the early 90s because the economic model of India changed from a quasi-socialistic model to a more free market economy, and because of that there was a sliding in the exchange rates, and so we went through a hard time, very similar to the just to relate for those of you who are in the younger age group here, what we went through about 6-8 months ago when the currency was demonetized. We went through a hardship period, but then things spring back to normal. I think Dr. Ramarao will remember the 1992 to 1996 was a fairly tough period in terms of grant funding. 2000, call it whatever, the millennium, the start of a new millennium, suddenly a lot of money appeared for research. And I would say we were beneficiaries of research grants which we sometimes did not know how to spend properly. Dr. Brahaspati was coordinating some of those programs, and I think it was very good that we had that kind of money because we could start new areas of research. As I said, at least my generation, we were not fortunate that when we started our career, we could think of starting an area which we thought was new to the department. So those who started their careers or those who felt they had still enough time ahead of them, and I think I was one of the beneficiaries of such, I would call as generous funding programs. We were able to get money to establish new areas of research, and the pharmaceutical solid state forms, that is polymorphs, co-crystals, salts, etc. I would say I was able to establish at University of Hyderabad thanks to funding which came from the Department of Science and Technology in the early 2000s. And I would say that really contributed, I think Srinivaslu, who's here in the audience, was one of the early students who, when he started his PhD, we practically had no major instrument in the department. And by the time he finished, I think we had all the major instruments in the department and my lab that would be, deemed required in any solid state laboratory anywhere in the world. So in a gap of 5 years, I think we could see the difference between night and day. And I think the generation of students who graduated during that period and after that perhaps had the right instrumentation in the right place to be able to carry out their work in the kind of topics that Gautam and Steve talked about. And many of the polymorph searches, except for, I would say, an automated high throughput screen, working manually as a PhD student, I would say they had almost all the analytical tools and laboratory facilities that one may need.
We know now that actually a human is better than a high throughput screen.
Yes, yes. I think we have some industry colleagues here who use high throughput, and they always say we still do a confirmatory manual screen.
Exactly.
Yeah, I think I see some of them there, they are all grinning. So the machine is there, but then the human hands, yes, you can't replace. Yes. So now fast forwarding to about post 2010, there has been a paradigm shift, and the paradigm shift is, I would say, global. In that increasingly, after spending a lot of money, I would say at least in India, in the last 3 to 4 years, increasingly this question is being asked that have we really benefited, and what is the benefits that we have from the increased investments the country has made in science, in fundamental research, in education, in higher quality of research, which is reflected in the metrics in terms of number of papers published, impact factors, number of researchers who are active in the country, and so on. Be that as it may, I think the bar is not the same today. It's a moving scale, it's a rising scale, and I don't believe that this is just specific to India. All over the world, I think governments and people are increasingly asking the same questions, and therefore a change in the model started happening over the last 5 years. In India, the major shift came through, just like you had your 1984 Act, we had the Knowledge to Equity Act of 2012, and Raspati knows about it, which was drafted by the DST, where academics till then could not start their own companies or invest in private equity. And post 2012, this became possible for academics working in universities, IITs, national labs, including the lab where I am now, CSIR NCL. Scientists can, be startup founders of companies. And this clearly changed the landscape, in that science to technology to innovation became a possibility for several academics, which it was not in the past. This also meant that we have seen, I would say, in the last 5 years, since I have been interacting with those who operate in the startup ecosystem. In the past, if somebody finished a PhD or a postdoc outside the country, he typically would look for a job. But in the startup innovation meets, which I have attended quite a few of them in the last few years, I increasingly meet young postdocs who start their career just like in the US, saying my dream is to, I have this lead or this idea, and my dream is to have a startup company. And we are seeing quite a significant number of researchers who are thinking like that. I will say the government funding bodies have also responded in a very positive way. The Department of Biotechnology has an arm which goes by the name of BIRAC, which is Biotechnology Industry Research Assistance Council, which is a side unit of the Department of Biotechnology. And its primary goal is to fund research which has the potential for technology and innovation, and then leading to product development. So rather than focus on just purely fundamental research, BIRAC aim is to fund startup companies or even established companies who want to collaborate with academic researchers. So whenever you have a bridge of a 1+1, that is one academic partner, one industry collaborator, BIRAC funding is a mode option which is available today. It was not there perhaps 5 years ago. In that sense, I would say the funding ecosystem to promote collaboration and partnership between industry, the government labs and industry and academic professors certainly has benefited through the BIRAC model of funding. As a startup company, which I talked about we started in 2010-11, we have been applicants and we were successful in securing funding for 2 BIRAC projects. These projects either are funded to the startup company or to the startup company in collaboration with an academic partner with whom they may want to set up studies either in a government lab or in an academic with an academic professor. So several of these startups, I would say, have flourished, have been successful. I don't have the numbers, but clearly the ecosystem today of startup industries who are founded by young PhDs or fresh postdocs is a fairly significant number, not as high as in the US, but the SBIRI grant, which is an acronym that is borrowed from the US, is there in BIRAC funding as well. So this is sort of the kind of background in which the 3 components, that is industry, academic and government labs, can find a way to partner with each other. When we talk of, now this is in terms of at a smaller level with startup companies. When we talk of larger level collaborations, which could make a disruptive difference, then I think one has to think of what would be the best areas in which these 3 sets of researchers could possibly collaborate so that you have a win-win partnership. Historically, we went through a phase where the DST was funding applied research, but it was largely fundamental in nature, and I think that phase seems to have ended by and large. Today, whatever funding is available in the name of partnership collaborations truly and largely is in the category which has the dual action of not only technology push but also market pull. It still needs certain refinements. It's a perfect model. It needs several refinements, I would say. And one of the refinements which, perhaps it is my phone, maybe thank you. One of the refinements which I feel, since I have participated in some of these discussions, is that even today I would say that the peer review system for even the projects which have the potential of translation and have to move to the market, the peer review system is still largely academics. Unfortunately, it should not be like that. It's largely academics. And so they are sometimes based on technology push. And there are many projects which have been funded by DBT, by BIRAC, which I would say score very high in terms of making a technological intervention. But the question is there a market gap that they are going to fulfill? And sometimes that last question, which I believe should be the first one, is not asked at the right time.
Ashwini, I want you to pose that question directly to Dr. Bharat. In terms of this mindset change, is there something fundamentally here that prevents us? This is a very serious issue, and it's a fundamental issue in this government academy industry collaboration. Because as long as the peer review system continues to be academically oriented or from the minds of academics, Steve, I don't know what's your experience in the States and things like that. people in the university actually said it is like a sort of anti-science or anything, and it's not required even. I mean, industry is there to do their thing. Why do you want to do a startup? Would you like to say something? But I want to hear it from the horse's mouth.
You will say, I'm a past horse. I'm super animated.
This is actually the good thing is, ecosystem is changing, and of course it's changing slowly. In fact, it's changing. But I cannot put it a little bit in my data. But the basic thing is, there is no compulsion for academics to collaborate with industry. Okay. They get their own promotion, they get their own this thing. In fact, we 10 years back, 15 years back, we organized in the green chemistry 3 workshops. In fact, Professor Venkata was there in workshop. One on cleaning the chroming industry. The second one was on bulk trucks, which was in building Hyderabad, ACT. Then the third one was pigment industry in Ankleshwar. Okay. All the 3 workshops, we have identified specific problems, issues to be addressed in collaboration with industry. The silver lining is industry was willing to support, and academic people partner was not forthcoming. In fact, we did so much of this thing. Of course, that was 10 years was long time now. The basic thing is there is no compulsion on the part of academia, academic people to collaborate with industry.
I want to ask a provocative question. Like, you say that there is no compulsion, but people could also argue that the whole idea of academics is that there is no compulsion to do anything. It's partly true, but see, on the other hand, on the other side of the coin, is it true that in chemistry many great discoveries came from the industry push, starting with Louis Pasteur's discovery of stereochemistry, and the Haber process came about because of the war, and something else. There was some push, there was some factor that allowed this very great discovery, scientific discovery, or are some very important scientific discoveries really blue sky? Is there something called a blue sky? I'm just throwing a number of questions to the people at the audience and the speakers.
I'll tell you one example. Bluetooth, who got the Nobel Prize for the discovery of Artemis. It was a directive from Mao
Yes.
to her, asking that she should find out cure for malaria. She is not an academician even.
I think she was.
She has not having papers.
Yes.
But she worked like hell till she came out with a compound what was being used and a natural product. In India, we spent huge funds, right from Sheshadri, Venkataraman, you name any unnatural one. But what we label ourselves, alkalides, phenols, if something different, they don't even touch.
But
How can you discover? We might have missed several biological active compounds.
But Dr. Ramarao, again, I'd like to give a counter example. When Todd went and asked, the young Todd went and asked Ruzicka whether that he wants to work with nucleic acids. You know what Ruzicka told him? He said these are not organic compounds at all. I mean to say, but your line of thought is very interesting because this sort of gives the feeling that all great discoveries have to be made with some push behind it.
That's right.
There has to be a push, otherwise you don't get that impetus or you don't get the spark.
One other good thing which is happening in this government is they are wanting, they are actually forcing us to go beyond discipline-based support to a theme-based support. Okay, for example,
Theme-based rather than subject-based.
okay, so
I would like to share 5 minutes.
Sure.
Let you come.
Yeah. No, we can have the comment.
Also, I would like to answer your push on behalf of the-
okay, that may have other side of it. That is of course the thing, but essentially things for people to think in a
I'll just finish shortly and then we can continue. I think the question as to why, what are the problems or what are the challenges that come when we have serious industry inputs in defining the problem for the academics. Professor Khandal is here, and I have sat in many meetings with him as the expert. Dr. Ramaswamy, who is former DST secretary, and also, I'll switch it on. Define in a very simple analogy what scientists in India and also scientists in the government labs, when they want to do an applied research, what they do, he says they will be working in an area, they will define a problem which is defined by them as a problem of application, and without asking anybody as to whether it is actually a pain for anybody or it fills a need, they will go ahead and complete it. So let us say you are in the business of making buttons. One chemist will make a green button, one will make a violet button, one will make a very large button, one will make a wooden button, one will make a button out of tin, somebody will make out of plastic. Then, rather than having asked prior to starting this project of button making as to who's going to use the button, he will make the button, he would have spent good amount of government money, and then he starts looking for who's got a coat in which that button will fit. So rather than look for the need and the application in the beginning, usually that question comes at the very end. And all I can say is, in the last 1 year or so, I have sat with Professor Khandal as the expert in several meetings, and this is the first question he asks in any meeting where there is a discussion on finding a product or a technology development which is going to fill a gap in the market. Where is the industry? Who is the end user? Have they told you that this is what they need? And if the answer is no, then please pause, step back, do that survey, come back with the data. Surely money is there, we will fund you. So to that an extent, I think the input from Dr. Khandal is extremely valuable, and to that an extent, it is making a difference in the way some of these projects are funded. But I would say in the early days, much of what happened was application in the name of fundamental research. It was just given a new name, and I think you also mentioned the same thing, Dr. Brahaspati, that industry wanted their problems to be solved, but academics were not coming forward to accept it or to adopt or take those projects, because many times, this is my experience, the problems posed by industry are not simple problems. They are extremely difficult, they are extremely challenging problems. Some of them do not have a simple answer for more than 30 years. For example, the low, I was familiarized with this problem, which is on tonnage scale on a daily basis, is this low density ammonium nitrate, which is an explosive. The high density polymorph remains as powdery form for a much longer time, but the low density, which has higher explosive capability, because of storage, because of being left in open sunlight, because of the shocks and jerks during transportation, it tends to form a cakey layer outside, and once the cake layer is outside, the inside may be powdery, but essentially that entire half a ton of that explosive becomes useless. It has to be sent back to the plant. They reprocess it. They just kind of recycle the drilling process, but there's a cost to it. So what industry would like is a low density ammonium nitrate form, not to form these cakes, and the caking happens because there is a transformation from the low density to the high density polymorph during storage because of heat. And so these are the kind of problems you pose to half a dozen people, even who are working in crystal engineering. they will say, yes, I can make a polymorph of the drug. But how to stabilize the low density ammonium nitrate? I think one of my scientists is sitting who came with me. Not an easy answer, because the answer is not at gram level. We saw the plant, it just runs 24/7. Several tons of ammonium nitrate is made every day, but it has to be stabilized, the low density form, because that's the one that has much improved explosive characteristics than the high density form. So some of the problems of industry are such that it's not, the answers are not easily forthcoming. And so probably what you said is absolutely right, that no, I'm almost done. No, Brahaspati, I was saying, yes, that problems were posed, but none of the academics came forward. Okay, so I'm almost done now. I think the opportunities for API generics, 505B2, 505J, they are certainly very fertile ground where all the three domains, the government labs, the academic professors, and the API generics industry, with no better location than Hyderabad, can find a very fertile collaboration and partnership. Just a few days ago, 9th of August, one of the statin drugs, Lupin, made the sodium salt as a new salt form under B2 classification and got approval compared to the calcium salt of the innovator. So just to mention that this is certainly an area in which one can explore future opportunities. I will finish with a small comment, which is again my own personal experience. On the slide which Professor Desiraju showed about 30 years of drug discovery, it also largely overlaps with my own professional career in India. Partly seeing several of the companies go almost to the peak of their R&D, including Dr. Reddy's labs in drug discovery area, and then deciding in a sort of gradual way to tone it down because they don't see the benefits accruing. Again, this is my personal experience and others may wish to comment on it. Based on the interactions I have had with some of my colleagues who have worked in two of the major innovator companies, namely Pfizer and Merck, the drug discovery program in these innovator companies is, the impression I get is, it is headed either by a medicinal chemist or by a pharmacologist or by a structural biologist who is trained in one of these areas, not who works in these areas, who has a PhD in one of these areas, or a combination thereof. But very rarely a drug discovery program in these innovator companies is guided in terms of which molecules to bet on or which molecules to drop, what they call as kiss or kill in industry. That decision is made usually by a non-chemist. And I'm not saying that chemists are not adequately equipped, but I think when a chemist adopts medicinal chemistry after completing his or her PhD, the rules of medicinal chemistry, the rules of modifying organic structures to give them the desired medicinal property, the nitro group example, and so on. There is a certain attachment to what chemistry is doable rather than what molecule is needed. And I know of one case for Merck where the head of the program, who was a medicinal chemist, not a synthetic chemist, basically said, I need this structure. You make it the way you like. I need this trifluoro group in this position. I need this chirality in this position of this nature. And I need the rest of the heterocyclic group on the right hand side. And I think some of you may have guessed which this drug compound is. And he told the synthetic team, it's your job to make it. In my vision, this is the drug molecule I want or something nearby. And then the synthetic teams had to find the right transformations, the right starting materials to put together the fragment, and I mean the rest is history. It's clearly a $1 billion drug of Merck on the market. In India, unfortunately, almost all the drug discovery programs, and I don't want to be misunderstood, are led by organic chemists who have, over the period, learned the rules of medicinal chemistry. So there is always this thing, if I think of this molecule, will it be makeable? And if it's not makeable, then there is a withdrawing of the target. Whereas for the structural biologists, or who may be doing fragment-based drug discovery of the type we heard from Tom Blandella yesterday evening, the method is in the background. The molecule is in the foreground. Whereas for the organic chemist, it's exactly the reverse, and that perhaps I think becomes the suicidal instinct for the medicinal chemistry program. You need someone who doesn't know much about synthesis. He knows the valency rules. He knows that this is a heterocyclic group. He knows a trifluoro group is needed at this position because there is a CH oxidation happening and I need to stop it in the enzyme pocket. He needs to know that much chemistry. But how to make it, he has an army of people who will make it. So thank you very much.
Friends, fellow chemists, scientists, crystallographers, maybe managers. I'm not a practicing crystallographer. When Professor Desaraju has asked me to speak on co-crystals and industry perspective, I told him I have never practiced co-crystal chemistry, but I am a fan of co-crystals. And I have been in pharma industry for more than 40 years. And some of my team members have been looking for opportunities to pursue co-crystal science. So I have accepted and given a title that I will do a manager's perspective rather than a scientist perspective. That of a SWOT analysis, strengths, weakness, opportunities and threats in the practice of co-crystals in industry. Professor Desaraju and Dr. Byrne have given excellent introduction and exposition of what co-crystals are, what the regulatory framework is, what the definitions are, what they can do, what they cannot do to industry. To my mind, co-crystals offer an opportunity for industry if what Dr. Nangia has said, Professor Nangia has said about how industry practices drug discovery research, he has given one perspective which I do not accept. That's not how it goes in the industry. But the co-crystal science has been recognized as an opportunity ever since industry made some success stories in polymorphs, the solid forms of drug substances. I call it a drug substance because many here are from Hyderabad or elsewhere in generic industry they call bulk drugs. But truly this bulk drug nomenclature is not used universally. My first, this is one. To put my talk in the proper perspective, I would like to define what drug substance, drug product mean. Drug substance means an active pharmaceutical ingredient, which people call it API. But when it comes to your filing an IND, API terminology is not used, it's a drug substance. It's nothing but the active ingredient, chemical substance, which imparts pharmaceutical activity in a medical preparation, medicinal preparation. Whereas a drug product defined as a finished dosage form containing a drug substance, containing API, with or without excipients. Got it? Okay. I have given here. Any substance or mixture of substances intended to be used in the manufacture of a drug product, and that when used in the production of a drug becomes an active ingredient in the drug product. You have seen drug substance is nothing but an active pharmaceutical ingredient. When it comes to co-crystal becomes a drug substance that goes into making the drug product. Here is another authoritative scholarly definition of a co-crystal. Swaradko, multiple component crystal in which all components are solid under ambient conditions. Here's the definition taken from Crystal Growth Structure journal. Solids that are crystalline single-phase materials composed of two or more different molecular and ionic compounds, generally in a stoichiometric ratio which are neither solvates nor simple salts. This is a much more elaborate definition, but the last one by Stanley perhaps is the most well understood by a common man. A molecular complex that contains two or more different molecules in the same crystal lattice. Here is a graphic representation of salts, co-crystals and polymorphs. Salts and co-crystals are molecular complexes, whereas polymorphs different forms or crystal structures of the same substance. Having defined these, we'll see these pharmaceutical co-crystals. The most important properties that you can see that they differentiate from a normal API is dissolution rate, solubility, chemical stability, moisture uptake. These physicochemical characteristics influence the therapeutic efficacy of many pharmaceuticals, all of us know. It has been recognized very early that physicochemical properties of pharmaceuticals can be improved or altered by obtaining co-crystals of a drug substance. Co-crystals incorporate pharmaceutically acceptable guest molecules into a crystal lattice along with the API. Coming to strengths, weaknesses, opportunities, and threats. Co-crystal technology as a tool to address chemistry and physicochemical properties problems in NCE, drug substance or drug product. Utilizing crystal engineering approaches and smart choice of co-formers to increase therapeutic bandwidth and outcome. Business opportunities offered by co-crystals emanate from the fact that co-crystals are patentable and therefore life cycle management of drugs can occur. In NCE research, even drug repurposing, a specific focus on neglected and orphan diseases can be looked at. The viability of approach to address unmet medical need also is a distinct possibility. What strengths the pharmaceutical industry can gain from co-crystal? As we said, first of all, co-crystals impart enhanced physicochemical properties, enhanced dissolution, permeation values compared with those of the parent crystalline pure phases. Co-crystal formation can act as an alternate to other solid state modification techniques such as polymorphs, salts and other things which we talk. Co-crystal can modify the solubility, permeability, which Professor Desraju has emphasized, and bioavailability of the drugs. In NCE research, co-crystals can be exploited as they can enhance blood-brain barrier penetration of drugs which are otherwise less permeable and therefore offer means of repurposing. NC attrition due to pure admin can be turned favorably. APIs and their important physical parameters, including dissolution rates and bioavailability, can be improved without compromising on the thermodynamic stability. Weaknesses are issues in reproducible preparation. They scale up and ultimately scaling up lab processes to manufacturing practices. Stability, integrity, and processability during formulation. Lack of clarity of crystal integrity in formulation is another weak point. In NCE research, repurposing drugs through co-crystal may not have a very strong IP and therefore cannot be exploited to the full extent. This is another weakness. In terms of opportunities, new opportunities of producing different, diverse solid forms of drug substances, repurposing of older drugs for other indications and for orphan diseases. In business, as we said, co-crystals are patentable and therefore they offer an opportunity for life cycle management. Consequently, intellectual property protection or a particular drug product can be extended. We expect to see more such examples as co-crystal engineering, co-crystal pharmaceuticals are approved by regulatory authorities. If you look at the opportunity, this is a vitamin D2, D3 co-crystal. D2 and D3, cholecalciferol and ergocalciferol, they are not stable. But once you see a co-crystal like this, it imparted stability. And this is something which you may see in practice, in medical practice in course of time. Co-crystals has active pharmaceutical ingredients. Fixed phases of defined stoichiometry. Co-crystals have different properties from the API. Therefore, you can distinguish and derive the benefit of a co-crystal superior to API. API co-crystal is typically fully characterized for solid state properties. Drug substance release and stability testing is performed on the co-crystal. This is perhaps the most celebrated example of a co-crystal that has been approved only in 2015. Incidentally, this is the first major co-crystal, pharmaceutical co-crystal that has been approved by US FDA who Novartis holds patent for this. Notice here, sacubitril, which is a neprilysin inhibitor, and valsartan, angiotensin antagonist, both were combined in 1:1 ratio and in that format. This is both a molecular co-crystal, it is also an ionic co-crystal and a solvate, hydrate. All three are combined in this. Fortunately, this co-crystal remained stable during formulation, and therefore, in my opinion, this has become a drug. If you make a simple co-crystal with a coformer taken from a gas substance, it may not impart this high impact. This is a combination of molecular co-crystal, ionic co-crystal, and also a hydrate. All three combined, it's fantastic. I think drug industry should look for opportunities like this. It will have a major impact and a big upside to exploit IP rights. No wonder this has displayed several surprising properties, such as intrinsic stabilization of the supramolecular complex by monovalent multi-coordinated sodium cations and water molecules, and it remains stable during formulation. Many co-crystal forms which you see in the table which I present, they do not offer these advantages. Among threats, physical combination of two drugs or a drug substance and a coformer. If it comes to business, different versions from regulatory agencies can preclude co-crystals as a viable pharma strategy. Now let's see, where do co-crystals fit in regulatory scheme? Salts, the same active moiety, but different API. A salt of an API can be patented, and it can be exploited. It has resulted in increase of solubility or imparted more stability and any other physicochemical characteristics that helped in granulation for formulation is a benefit that is recognized in awarding a patent. Co-crystals is a new regulatory class of solids needed. There is a question mark. Yes, in my view. Positioning of co-crystals as drug substance, that is European Medical Agency recommendation. FDA's definition implies co-crystals as dissociable API-expand complexes, blurring the boundary between co-crystals and physical mixtures. And more importantly, the defining of co-crystals as DS or DP. There is an uncertainty. Steps involved in preparation of pharmaceutical co-crystals. Bulk drugs or API, synthetic organic chemistry is the backbone of process development. This is highly evolved. The generic industry in Hyderabad, which is bulk drug capital of the country, thrives because of process chemistry. In the case of co-crystal, it's a different game. In bulk drugs, even 10-15 steps synthesis is nicely practiced, scaled up. Streamlined processes meet all regulatory requirements. But in the case of pharmaceutical co-crystal, the technologies are not yet so streamlined, not yet automated. Very often batch processes are now converting into continuous processes in bulk drug industry. First you have the selection of an API, which is a fit case. Selection of the co-former. It can be a drug substance, another drug substance, or it could be a grass substance. Co-crystal formation and screening should be done in laboratory scale. These are very primitive methods, not as well defined as organic synthesis. Gautam Desraju might get offended if I say this. A co-crystal characterization. This requires high-power crystallography. With crystal engineering even to create co-crystals. And finally to study the co-crystal stability. It has to withstand the formulation processes and protocols. During formulation, the co-crystal stability and its integrity may be maintained. Well, 5 minutes, yes. Pharmaceutical products containing co-crystals. Major pharmaceutical co-crystal patents issued and drugs based on pharmaceutical co-crystals as APIs made their way into clinical trials. There are a number of co-crystals are now in clinical trials. Pharmaceutical co-crystals also resulted in many patent applications, but only a handful of co-crystals have entered into clinical development. Approval of LCZ 696, that is the interest of Novartis, may be an indicator for additional approvals to be witnessed in the coming years. Indeed, pharmaceutical industry must exploit situations that will derive co-crystals which fit into this class. They are molecular co-crystals, ionic co-crystals, as well as solvent. Sacubitril did not become a successful drug. Valsartan is used, but very limited among all the angiotensin antagonists. But the combination in that crystal form, co-crystal form, is a big hit. This is going to be a blockbuster. The pipeline of drugs in regulatory review will continue to use mix of technologies. Co-crystal formation is one such. Industry will certainly woo this area. Some of the pharmaceutical co-crystals in potential development pipeline are presented in the next few slides. There are a number of them. Carbamazepine Professor Byrne also has presented this. This is a drug discovered in 1953 for epilepsy and bipolar disorders, but now it has been co-crystallized using coformers such as saccharin, nicotinamide, and salicylic acid. The advantage is improved solubility. As it has been pointed earlier, most of the drugs that we handle today suffer from lack of sufficient solubility. So co-crystal formation is one such remedy to impart greater solubility. If fluoxetine hydrochloride, another CNS drug, this was also discovered in 1992. It is still used in practice, but look at co-crystals with benzoic acid or fumaric acid has again brought improved solubility. And this development took place in 2004. Obviously, there are some more years before it can become a drug, provided it remains stable during formulations, clinical trials, so on and so forth. Itraconazole is another example. Modafinil, all these higher oral bioavailability and physical stability in one case, solubility, dissolution, bioavailability, and dose response. Developed only in 2009. You see, all these were developed prior to 2011. In 2011, US FDA has come out with criteria for approval of these. So we have to see approval status in course of time. Here are some more examples. Celecoxib is one, developed in 2012 as a co-crystal. Tramadol, discovered in 1977, may be resurrected using a co-crystal. The advantage is a increased aqueous solubility. Entresto, which we have talked, that is, this compound. OALG is in a recent journal, Drug Discovery Today. The number of publications that are coming out each year are increasing. This means a trend towards practice of co-crystals as a means of repurposing, bringing back failed drugs. We may see such a trend in course of time. Reformulation of existing drugs and improved performance, life cycle management, enabling development of known compounds into novel entities that are patentable, exploit co-crystals for non-obvious surprise properties like Entresto. Pharmaceutical co-crystal technology is useful if one identifies and develops new proprietary forms of widely prescribed drugs and offer a chance to increase the number of forms of an API and their therapeutic use. So in conclusion, I must say, there exists a strong need for streamlining co-crystal technology and regulatory perspective. If I may go back and say, a salt of a drug substance also can be patented. but it can be easily made, scaled up, manufactured in tons. Salts are well defined in terms of, it's a theory. Acid reacts with base to produce a salt. Whereas in co-crystals, you have to select a co-former. You have to first experiment and observe to say that it formed a co-crystal. It's still empirical. There are no ground rules and guidance factors to which co-former should surely form a co-crystal with a drug substance. But if my personal, I'm not a practitioner, I'm not a crystallographer. I would like to advocate to the industry that co-crystals of two drug substances, such as what you saw in Entresto, is more useful than producing a co-crystal with just a co-former drawn from list of grass substances. Thank you, sir.
Many questions which provoke further thought. And in the interest of time, we must move on, but I can't help but saying that co-crystal development is our solid state equivalent of process chemistry. What was done in the past for molecules, we sort of getting to the same thing using a different route, but now we must think supramolecularly in this process. So it won't be a strict analogy, but the parallels are many. And I believe that in the same way that India seized this opportunity of process chemistry at a time when it was badly needed, co-crystal technology is filling that gap now. And yes, I think this talk raises many questions. And we'll move on to you and finally you before we conclude the session. Thank you very much,
Just
Dr.
one comment, sir.
But
Co-crystal technology is only to extend the life of a substance which is already proven to be a drug. So it's not going to be like an NCE discovery.
no, it's not.
It's an innovation.
It's an innovation.
Although I might add again provocatively, when you gave the Entresto example, it looks so different than you. I was actually wondering, is it like an NCE almost? Because drug co-crystals may actually be almost quasi-NCEs,
Whatever.
and it's debatable.
It's not like there are so many other combination plants, dosage forms. They are not co-crystals.
No, I know. I know that. But it's still it's wonderful when you have a topic which is worthy of fundamental research and at the same time is also worthy of quite heavy academic applied and industrial type research going on at the same time. So I think the communities have a lot to learn from each other.
Yeah.
We are both sort of groping a little bit, but I think we are groping to the same. So I think it's a good opportunity for synergy.
Thank you, sir.
Thank you very much. Professor Mithai, you are anxious that he's such a so many commitments as the president of the company that even getting away from there for 24 hours was a challenge for him. And I deeply appreciate the fact that you made it possible to come here. And now we come to somebody who is now deep in industry. So this academic team, government and all we have, although he talks to academics and talks to government people, but he will now give us the perspective from the industry side actually.
Thank you, sir.
This is the real world.
Thank you, sir. In fact, I was anxious not to come first on the podium, anxious only about the fact that by the time my chance comes, there will be very small audience. That's what was worrying me. Rest all is secondary for me. So I am happy they are there. परम आदरणीय प्रोफेसर देसी राजू जी, देसी राजू गारू, Adharaniya Professor Venkateshwarlu ji, Professor Ramarao, Brahaspati ji, Professor Nangia, and Honorable Professor Steve Brand. Let me tell you, it's a wonderful delight for me. The other day, when I met for the first time with Professor Desiraju, I was not very sure how my meeting is going to be in times to come. But as I am the believer of Indian philosophy, that you always meet good souls to meet again. So that was my first meeting with him. And I was so damn impressed with him that how can a professor from Indian Institute of Science, Bangalore background can be talking something highly practical, can be talking something, because in India, and let me tell you, my presentation is made by one of my students, but now I don't feel like talking about that. The reason for that is, this session, I'm very lucky that I came in the last to speak, because so much of knowledge that I have gained. And when they were talking on the subjects which are very close to my heart, but I never worked on them so much as they have worked, I was trying to interpret and use the knowledge that we have been dealing with, or we have been creating on our day-to-day work in industry life. So the meeting with him for the first time was real delight. And after that, it's all irreversible. The other day he called me and he said that you should come and give a lecture, give a presentation in the international conference. I thought this might be an international conference like any other international conference. And when I looked at it, crystallography, I said, dur ka mera, I don't have any connection with it. What will I talk, on which topic I will talk? Because let me tell you, what all I have heard so far, the solubility, the permeability, the bioavailability, pharmacokinetic, all parameters. This is what we are doing from the other side, because we are the largest manufacturer of excipients. We are the largest manufacturer of surfactants, emulsifiers, dispersants, suspending agents in the world. And in the world, when I say, not in terms of quantity, but in terms of the quality, in terms of the nature, our products are derived from natural resources, and the natural resource, our raw material is molasses, sugarcane molasses. So always we have been trying to develop products that can make anything insoluble, soluble. And here I am subjected to crystallography, where, because, and medicine's crystallography, I was really wondering. But then I listened to Professor Steve Brand, who very nicely presented and elaborated the importance of crystallography from the point of view of regulatory agency. I mean, you can really catch hold of a manufacturer of a drug, whether it is a monohydrate or a dihydrate. It is just like gypsum or calcium sulfate of what form of number of molecules attached with it. It's very interesting. We don't look from that angle. You understand? Then, of course, the presentation by Professor Nagia, who talked about the DST and talked about the professors getting grants. And then, of course, the DST, how they do the peer review. The chairman of the committee asked his all friends, where they are the chairman. So they ask their colleagues to, because I am the chairman of this committee, so ask your students to put the papers for grant. So you pat my back, he will pat your back. Okay. So you sanction their project. When your students go there, and fellow like me will never be in the committee in DST. Because they did the danger of keeping me in 4 or 5 committees about 10-15 years ago, when Dr. Ramamurthy was the secretary. He said, yeah, fellow like you should be there in the committees of DST. I said, okay, no problems. After some time, all the professors started saying, please let us know when he is not coming, then only we will come to make a presentation.
Professor Kandal, I too chaired a PhD in DST 20 years ago. And the rest is history.
So this is so very interesting listening to Professor Nagia. And he had said kind words for me. I am really obliged. And thank you. Whenever somebody appreciates you, including when claps when you talk, it's like blessings, okay? The souls give blessings. When I was listening to all these speakers, I was just wondering, and let me tell you, when I have to give a lecture on medicinal chemistry and medicines, I really feel very bad. You know, why the hell the human being should take medicine? Can you believe it? I never take medicine. And I will never take medicine till I die. I will go by the philosophy of Jains, that the day I decide that the Atma has to leave this body, I will resign myself. The Brahmacharya, Grahasthi, Vanprastha and Sannyas, and leave this world. Why the drug? Then the second thought was coming in my mind, why the drugs have to be formulated in the form of capsules, in the form of liquids? Well, these are the thoughts which I was, when every time each speaker was telling solubility, permeability, and can you imagine? Permeability is one word. One of my students did PhD on development of membranes which are semi-permeable. And you know how this was? This must be co-crystals and crystals. I never looked into from that angle. I was only looking at the molecular mass that they were creating. And they were all elastomer molecules. Amides, polyamides and elastomer. And that too soluble in water. So this is a story. I was just wondering, our forefathers in India, you know, the Indian Saj, Professor Steve Bryan, please carry this message back home. They keep doing crystallography, co-crystals and salts on these organic molecules. They never bothered to think about Bhasma. You know, for about 10 years, I had 3 students of mine who were trying to validate a process for manufacture of Bhasma. Can you imagine how the Bhasma is made? Professor Steve Bryan, have you heard of Bhasma? I don't know whether it is ash, or it is co-crystal, or it is crystal, or it is complex or supramolecular. You know, each element, each component of it is being treated. It's a process highly, highly sophisticated. And it's meant for diseases which are incurable. I remember the case, a fellow who had a heart problem, and the doctor said that you should not eat desi ghee. Okay, and you are not supposed to do this. Desi ghee is the, you know, cow butter oil that you call it in US is desi ghee. We do it in our own style and we cherish it. When we stopped eating desi ghee, all of us, we became sick. You know, in India, as long as we were vegetarians, we were not sick. The moment we started eating animals, all these problems started, then we went into drug discovery. I really wonder when somebody says drug industry is growing. I say, how drug industry is growing? No, per capita consumption is increasing. That means your people are becoming more and more sick. What the kind of things you are talking? Smart city or drug industry growing? I always tell my students, teachers and everybody, wherever I go to give lecture, imagine 1,250 million, okay? Like Modi ji says, 125 crore, mere 125 crore Bharatiya. Imagine they don't fall sick for 1 year. You know what will happen? It will be $1 equal to 70 rupees, no. 1 rupee equal to $70. Can you imagine? Have you thought of it? If all we Indians don't fall sick, all the medicines that we produce will become so cheap, we will export all, like China exports to all over the world, we'll export it to Africa. All our hospitals will welcome people coming from New Zealand, Australia, Belgium. We will treat them because all our doctors will be busy in treating them rather than because all of us are healthy. And it is so simple to remain healthy. It's so complicated to become sick. And it is so simple to become more sick. Okay? Because the moment you take one medicine, you have already registered your name for another medicine. And many a time wonder when I was listening to all these lectures, I was just wondering, any organic molecule, if it is tested at ppb, ppt level, maybe it's a very good medicine. Let me tell you, all this drug discovery is really very, very scary. Because you tell me one drug substance which has not been banned. The moment it is discovered, already its Janampatri is written that by this so many years it will be banned. What is all this going to happen? What is all this happening? Is any of the Ayurvedic product banned? Tell me. Thousands of years ago, Ayurveda was developed. Just now somebody gave the example of Artemisinin, the Chinese fellow. You know how that Nobel Prize was given to that person? The person was working on real Ayurveda, concept of Ayurveda, and ended up developing Artemisinin. When I asked my students to prepare presentation for me, the topic was success factors for drug industry in India. You know, the latest trends, when I was told that latest trends in the world is that prepare a medicine suiting to an individual. That is only happening in India, because you go to the Vaidyaji. Vaidyaji will prepare a formulation for you, specific for you. He will check your nerves, okay, your pulse. He will check your tongue. He will check your all the details of what time you get up, what all you eat, when do you have your dinner, do you freshen up properly, do you have this Kapha, Vata, Pitta. This Kapha, Vata, Pitta we are reaching to. I am telling you, very soon, in next 50 years, the Americans, the EU and Japanese will be talking of Vata, Pitta and Kapha, which our forefathers had described long ago. Let me tell you, if Indian drug industry has to progress, it has to do differently. Whatever is going on, continue with this. Dr. Venkateshwarlu, this is for submission to you. Ye aapko samarpit hai. Whatever we are doing, continue doing it. But start a new stream. I just wonder why drugs are not eaten. Why the hell you have to formulate? Why they are not eaten? I was wondering when I was listening to all this. One example he gave, that the drug was so bitter that he could not, gulp it, so it need to be formulated that way. All our Ved, they used to give, this is to be mixed with shahad, this is to be mixed with milk, this is to be mixed with each drug. The Ayurvedic drug preparation was supposed to be taken by mouth, contact with the mouth, because that is where the bioavailability enhancement happens. Nobody looks into it. Nobody considers it, because now it is good that the Europeans and Americans have started talking about integrated medical science. They say also learn from Ayurveda, but in India we say no, Ayurveda, this is separate. We are MBBS. Okay, because MBBS means if you are taking the medicine of Ayurveda, don't come to the hospital and don't blame me tomorrow. I mean, they give when a woman gets pregnant, what does the gynecologist say? Oh, you have a problem, you should start taking protein, you should start taking omelette. The lady says, but I am a vegetarian. No, you see, if you want your child to be healthy, it begins from there. The child is born, then, he may not have eyesight issue, you should start giving him omelette. A fellow like me who is a strictly vegetarian, okay, who has no distant relatives also non-vegetarian, he is made to eat non-vegetarian, inviting problems. How the mad cow disease happened? Because the cows were given the meat to eat. The cows is supposed to be vegetarian. Okay, another thing, these polymorphs. My students, when they used to work on polymorphs, we used to only work on the food products, because one group was working with me on food processing. Why a particular food with the same ingredients tastes different is because of the polymorphic form of the same product. How the starch is, how it is, desi ghee mein kaisa swad aayega, tel mein kaisa swad aayega, aur swad se kaise uska bioavailability pe effect aayega, how it will be good. You never, for example, you never eat methi. When you have a stomach problem, gastro problem, you are advised to take methi. They will never ask you to chew the methi. They will say you gulp it along with the water. You must have done it. Sometimes they say do it along with salt. Let us take example of toothpaste. All over the world, the toothpaste is also a pharma products. We call it oral care. Okay, it's not pharma in the sense that it is a medicine, but in fact it is worse than that. All over the world, the toothpaste is sweet. Can you imagine? And what do they add for sweetening? They are adding saccharin. All over the world, saccharin. They are not adding sugar. If they add sugar, I'll be very happy. But they are adding saccharin. If a child, and what do we advertise? We advertise, it's very sweet. So that the child is very happy to do toothbrush in the morning. Because he enjoys, half of it he will eat and half he will use it for cleaning the teeth. It's happening. And then we complain that in India we are increasing, the number of diabetics are increasing. Because you are giving saccharin, you do the daily intake, calculate the daily intake of saccharin. And saccharin is carcinogenic. At one time it was banned. You can check it. Nobody talks all this. And in India, those who are teaching, they don't do research. Those who are doing research, they don't develop technology. Those who are doing technology, they don't teach. I had made a presentation, but I thought I will not make this. I should share with you something which is very, 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 very basic. I'll tell you how. We say, if you are a teacher, you are supposed to be PhD. If you are an IS officer, you are just BA pass, graduate with political science. You know, when these days you visit houses and you say, beta, what would you do? No, uncle, I am preparing for IAS. So for IAS, no, I don't want to do science, commerce and all that. So what would you do? No, it's very easy to clear IAS by doing political science. Very good. So he will study the worst of the subjects which have nothing to do with humanity. And he will decide all the policies meant for humans. After graduation, he will become an IAS officer. Indian Administrative Services, he will become a bureaucrat, top bureaucrat. He will become the Chief Secretary of Andhra Pradesh or Telangana or something. He will take a decision whether Telangana should have this kind of farming system or this kind of drug industry, what incentives to be given, he will decide. And that fellow who is an engineer, he is supposed to do, as per AICT, supposed to do BTech, MTech and then PhD, and then he can become professor. What silly thing. Another beauty. When you don't get anywhere any job, then you become a teacher. Why you are a teacher? No sir, first I try railway board, then I did not pass, then I tried probationary officer, then my age was crossing. In the meantime, I was continuing doing my PhD. So my guide said, why don't you become JRF, SRF? And then I became, so this, sir, I became like, so why you became assistant professor? Sir, it is very easy. 8 years. And like I became, when I became first time, I was 24 years, 5 months old, youngest teacher of a central university that time, in 1982, in Indian School of Mines, Dhanbad. I became lecturer. So we used to discuss, oh, so in 18 years time, we'll become professor. So you know what was the keenness? So, but 18 years is too long, yeah. I should become professor earlier. So after 3 years, I left university. I went to public sector. Then I said, public sector is no good here. I should go to Indian industry. You know, multinationals I should go and see how multinationals are working. So I went to multinational. Then I found, no, this is no good. Came back again to R&D. I tell you, the whole thing is what we are supposed to do, we don't do. A teacher, now they have come to the conclusion. Now look at this. Universities don't have students for admission. One thing. But all the coaching centers, they are flourishing. Students from Andhra is one state which has maximum number of students trying for JEE in IIT, NIT. Okay? Maximum number of students followed by UP or... Sir, khatam karte hain aap kah do.
I wanted to add a story to
हाँ जी sir. हाँ जी sir.
See, this is going slightly in a different way, and I would say that as a former president of IUCR and having attended many IUCR congresses, I have not often heard a lecture like this, but he does address certain very basic issues that are beyond, I would say, the wisdom and depth of this particular micro-symposium. But in the end, I mean, at the root and thread of what Professor Tandon is saying is that there must be an element of practicality in what we are doing. And I do feel now that very happy here, Steve, because there are a few foreigners here. But we are addressing issues that primarily are connected with Indian industry, Indian research in an area where India is particularly strong. And I think the essence of, again, what Professor Tandon is saying is that each culture, each country should play to its strengths. The moment you start trying to aspire to playing to other people's strengths,
You will be lost.
you will be lost. You will be
Absolutely.
lost. And yes, and he says it with nobody cares about academics anyway, it doesn't matter.
But sir, this time I'll be allowed extra, no? Which you are taking.
No,
Which you are taking.
because we have another meeting.
Sir, I'm coming to the point. This was a background which I was talking. This was a background. Okay. Now,
but no, your background is better than your slide. You've told us something rather essential, no jokes apart. I would take what Dr. Khattar says somewhat seriously. All of you, young people, you are in industry, aspiring to get these industry jobs, which all of you will very easily get. Because in crystal engineering and industry job, the connection is almost immediate. But you must have, take the essence of what Professor Khattar is saying. The rasa, as it were, the essence. Take it and put the element of practicality into your research, because India is a country with big problems. And very many times in India, when we have been successful, we always don't fail. Let's not be very harsh on ourselves. When we are successful, we have been able to adopt an intermediate technology that bypasses many standard routes which the Western world has developed.
sir, 5 minutes you give me, I'll finish my lecture.
Finish. I'm giving you exactly 5 because
5
we have
minutes.
to go to the other
No, I
meeting.
also not keen to continue. Listen one thing. Do we have a course called BTech Crystal Engineering? We don't.
No, we don't.
We don't have. Why? Who will make the syllabus for this? Who will make the curriculum for this? Those who are doing in the industry, they are supposed to write the curriculum. They are supposed to teach. They won't do it. They have no interest, or maybe they are not told, or they are not invited because somebody has to invite them. Our system is such that we do not involve people who are supposed to be knowledgeable, who are knowledgeable. We don't have a system that we use their knowledge. The knowledge is not coming into the stream. How will the knowledge be created? The addition of knowledge, the value addition of knowledge is done only once you create knowledge. Now, in my presentation, I had a lot of slides, but 2-3 slides I want to show it to you quickly, so that because the chairman sir is so kind that he has allowed me 5 minutes. So I'll just show you. This would be required to develop one molecule. Very simple answer Dr. Venkateshwarlu has presented, that what is not useful can be made useful if you understand co-crystallography and co-crystal manufacturing process. But what I am trying to show here is the crux of it. Let me tell you, people say that materials is the key for drug industry success, because we are heavily dependent on intermediates from China. Let me tell you, if China does not, we stop taking it from China, Chinese industry will close down. So we should not be scared of it, number one. Number two, let them manufacture the intermediate and pollute their own environment. We get the intermediate and value add it. There is one slide which was there in this, I don't want to go back to that. We import X amount of drug intermediates and drug, and we export 5X. So suppose some figure was there like 4 billion we import, 20 billion we export. So this is the best thing Dr. Venkateshwarlu. We should do it. But the success is that we are supposed to do materials proper crystallization, proper characterization. And all that is necessary we should be knowledgeable about. Now here in the materials I am just telling you, it is not that a particular molecule can be done only by one route. The second thing is, you have a drug substance and drug product, and then you search its excipients. In Ayurveda, you know already a drug substance which is along with the excipient. You don't even have to search the excipient. Here the work is done to see whether the excipient is useful or not. You know what is the US pharmacopoeia, British pharmacopoeia, Japanese pharmacopoeia talk about the excipient? It has to be neutral. It is not supposed to be reactive. It's all nonsense. Why it should not be reactive? It is going to the body, it should enhance. It should enhance the bioavailability of the drug. That happens with the Ayurvedic products. Why can't we come out with one Ayurvedic product with all that is being done by the allopathic drugs? Why don't we take this as a resolution in IISc, in CSIR labs? Why do we have to go for a molecule development? I fail to understand. जो चल रहा है वो चलने दीजिए. Make money, continue with that. But for the transformational thing, have a look. Give a pause, have a look and start thinking in a manner. 125 crore. 70% of them are still dependent on Ayurveda. Whether it is Telangana or it is Jammu and Kashmir or it is Arunachal Pradesh. Everybody, if you have a problem, the advisor is there next door, your neighbor. He will say, you take tulsi tea. He will say, no, you take neem flower, add in this, and or you eat halwa. You will be perfectly okay. Or you eat vada with that ghee on that. You understand, so much of knowledge is there. That knowledge, if we utilize, I tell you, all these fellows whom we follow, they will follow us. Second thing is the product. The product, proper, we have already seen whether it is a salt, which salt. And let me tell you, a given salt, which is defined by organic chemist and by solid state chemists, if they say it is not soluble, it is not permeable, a surface and colloid scientist like me can make it bioavailable, can make it soluble, can make it permeable. You need to modify. When you can modify, you can create nanomaterials by doping and all that. Why can't you do all these materials by doping surfactants? You have so different kind of surfactants. You pick up any insoluble substance, you just coat it with surfactants and it will very easily go into it. Or make capsules, like these days we already have formulations in which we have concentrated, encapsulated, concentrated suspensions. Or encapsulated, which get into suspension form the moment we take them. Similarly, processing and biopharmaceutics. So with these words, friends, I was supposed to have spoken 30 minutes, but doesn't matter, I don't regret it, because as long as I see your faces happy listening me, my job is done. My life is done. Thank you very much.
Thank you all very, very much.